Hello, welcome back. This is Professor Young, and we are beginning our look of how we go from DNA, and specifically what is DNA as well, but how we go from DNA to this concept of proteins. We know and we understand that proteins are vital to almost everything that happens within the body. But what we have not quite handled yet or tackled yet is how. How do we actually make proteins? What is the factor that oversees when we make proteins and when we don't make proteins? And so with that said, the next couple slides are going to kind of set us up with an understanding of first what proteins are, what DNA is, what DNA is made of, and once we go from and have an understanding of that, then we can kind of go into beginning the journey of really looking at what a protein is and how it's made. And so that's kind of where we're going over the progression here. And so, starting at the basic, we know a couple things already about DNA. First of all, we understand that it is a nucleic acid. We also know that it is one of two nucleic acids that we have within the body. We know that we have both DNA and RNA. And again, both of these are types of nucleic acids. We know that also that the nucleic acids are made up of basic structures or units known as nucleotides, and that nucleotides are made up of this concept of a sugar backbone, which in DNA is going to be deoxyribose, and in RNA will be ribose. It is also made up of a phosphate group, and it is made up of a nitrogen base. Now, it is the nitrogen base that we are most familiar with, or need to be most familiar with and concerned with as we go forward, looking into how exactly DNA copies itself and how we go from DNA to RNA to proteins. So it's these nitrogen bases that are going to be vital for our understanding. In DNA, we have really four base pairs. Adenine and thymine are always going to be bound up together, and guanine and cytosine will also be bound together. And again, adenine is associated with DNA, guanine is associated with DNA, cytosine is associated with DNA, thymine is associated with DNA. The switch comes in here because we also have uracil. It is kind of the odd man out in a sense here. And I apologize if the audio is coming in and out. For some reason, I have a crappy connection going on here, and so I think I keep losing signals. But I apologize up front for that. The uracil is a part of RNA, and we also see adenine being a part of RNA. We also see guanine as being a part of RNA, and we also see cytosine as being a part of RNA. Thymine is not a part of RNA. And so literally what happens is when we make RNA, the thymine is replaced by uracil. And so uracil will then go ahead and bond with adenine instead of thymine. So the adenine is binding with the uracil in RNA, the adenine is binding with thymine in DNA, and guanine is always bound with cytosine. And so exactly what we just got done looking at here, again, this here is just simply the subunits that make up a nucleotide. And so if we look at these entire structures here, all three of them, this here being the phosphate group, and this here being the sugar, and this here being the nitrogen base, all three of these are going to come in together to make one nucleotide. Now, how do we know this is a nucleotide that is associated with DNA? Because after all, we've got adenine. Adenine can be both adenine can be both DNA or RNA. We have a phosphate group, which is very consistent with both DNA and RNA. And so what sets this apart, the way that we know that this is a DNA nucleotide and not an RNA, comes in right here. We can see, hopefully at least very clearly, that if this was a RNA molecule, that we would have an oxygen that would be inserted right there. And so if this was RNA, we would have a completed hydroxyl group in that 2' location, what we would refer to as what we would refer to as the 3' location right here. And we don't have it. Right? That is non-existent right there. Right? And so we only have an H, therefore we are missing an oxygen, deoxyribose. Our bases, our nitrogen bases, can be broken up into two major overarching groups. We have both the pyrimidines and the purines. Purines are up top here. Pyrimidines would be the guys right down here. And you can see that there is a major difference between the two of them. Adenine and guanine are both considered purines, and they are double-rung structures. And again, I think you can see that relatively clear. Here we have one rung. Here we have two rungs. Here we have one. Here we have two. Right? And so we have a double-rung structure for our purines. Adenine and guanine are the examples of that. And our pyrimidines are all made up of single-rung structures. And so cytosine only has one rung, thymine only has one rung, and uracil also only has one rung as overarching structure. The other thing that we can tell with this, or the other thing that we need to understand with this, is that cytosine is always bound with guanine. Right? Cytosine is always bound with guanine, and guanine to cytosine, and it is a triple bond. It is a triple bond. And we also see thymine that is always only bound to adenine. And that is always a double bond. Adenine to thymine is always a double bond. Now, this is assuming that this there is all being done in DNA. In the case of RNA, now we come down to the uracil, and uracil will bind with adenine in the case of RNA. In the case of RNA. And that's also going to be a double bond. That's going to connect the uracil with the adenine. And so putting this all together, here we can see that we very clearly have DNA. We know that it is DNA because if we look right here, we can see that adenine and thymine are bound together, and that only happens with DNA. And the other thing that we can see very clearly with this diagram is the fact that adenine and thymine are going to form double bonds, whereas guanine and cytosine are going to form triple bonds. Right there, and so you can count the 3 bonds between the G and the C, and the 2 bonds between the A and the T. And so when we go to go ahead and make a copy, when we go to replicate RNA, when we go to replicate DNA, my apologies, which is what this is really called. This is called DNA replication. In order to do that, we need to break the bonds that are holding the base pairs together. And in order to do that, we need the assistance of an enzyme. And the enzyme that is responsible for unzipping the section of DNA that needs to be replicated is DNA helicase. So DNA helicase is the enzyme that is responsible for separating out the 2 strands. And when it does that, we of course would have an A, T, C, G, G, C. And that would make up 1/2 of the strand. And then counter to that, we would also have a T, an A, a G, a C, a C, and a G. Right? So we have these 2 strands that have now separated. They are separated because of DNA helicase. And these 2 strands are derived from the parent strand. And both of these, both the first strand and the second strand, both of these guys are known as the template strand. Both of these, because they're going to serve as the blueprint for how the new strands of DNA are going to look. And so there is a second enzyme that now is going to come in, what we refer to as DNA polymerase. And DNA polymerase is an enzyme that is going to basically bring in the complementary base pairs. We're going to bring in the complementary base pairs. Complementary base pairs. Complementary base pair for adenine is thymine. The complementary base pair for thymine is adenine. The complementary base pair for cytosine is guanine, and the complementary base pair for guanine is cytosine. And so wherever we see the A, we can expect that what is going to be bound to that is going to be a T. And wherever we see the A, we can assume that wherever we see a T, we can assume that an adenine is going to be bound to it. We see the cytosine, guanine is going to be bound to it. Wherever we see guanine, cytosine, and guanine. And we can also come over to this side, to the second template strand, and we're going to do the exact same thing. And so we have a T there. We're going to have an A that is bound there. And we have T. And so now that our new strand that we've just created based off of the template strand is there in green. And then, of course, now that's DNA polymerase. DNA polymerase brings these new nitrogen bases into and matches them up with their complementary base pair. And then we have yet another enzyme, and that would be DNA ligase. DNA ligase is the enzyme that is going to ensure that we have re-zipped our strands of DNA. And so wherever we see the A and the T, we know that we have a double bond. And so there would be a double bond here. There, wherever we see a G and a C, we know that we would have a triple bond. And this would be done for both strands, both newly formed strands. Just like that. And you have yourselves a completely replicated strand of DNA. We started with one being right here. And from that one, we've now created two. There's the first new one. There's the second new one. And notice that they are both identical to the original. That's going to come into play a little bit later. Not yet, but it will come into play. So that's DNA replication. That is DNA replication. One other thing about DNA, before we jump ship and go to RNA, which is going to be a little bit more cumbersome, and that is, I should probably mention a little bit, at least a little something on the structure of DNA. And really what we see here is we see three different stages of the exact same structure. All right. And they would be chromatin, chromatids, and chromosomes. Now, as I was explaining in class, it's kind of like if you take Christmas lights or holiday lights, and they've been shoved up into the attic all year long. You didn't put them away properly at the end of last season. And so when you pull them back out, they kind of look something to the extent of this. They are just in one great big mess. Not going to be able to do much with that. Well, that's exactly, for the most part, what chromatin looks like. Chromatin is a dense ball of unorganized genetic material. It's a dense ball of unorganized genetic material. It's very stringy in appearance. And we'll discuss where that fits in the greater scheme of things here in a little bit. Chromatid, on the other hand, what ends up happening here with chromatid is this tangled ball of thinned filament-based genetic material. Eventually, condensed And when it condenses down, it actually coils itself up, like such. So it takes that long, stringy appearance, and it condenses it down to this right here. And so we end up with something a little bit more organized overall. And again, that is a chromatid. That is a chromatid. Chromosomes are produced when you take two identical chromatids, what we refer to as being sister chromatids, and you link them together. And so we would have to come in here and produce another sister chromatid. And that sister chromatid would be lined and matched up with the first sister chromatid. So the joining of two sister chromatids forms a chromosome. Right. Now, typically what happens is biologists get lazy and the general public gets lazy, and we kind of consider all of these to be chromosomes. In the strict sense, though, there is a very big difference between chromatin, a chromatid, and a chromosome. And so there are huge differences there. There are huge differences. Now, one last little tidbit of information, and then we're going to jump into the fun stuff, that being RNA. When we have, and I should say, as we do here, when you have two sister chromatids, there's sister chromatid number one, there's sister. Both of these chromatids are connected to one another, and they're connected right here in that region, and they basically form a bond where the kinetochores are. Now, the kinetochores are these kidney-shaped structures that you're looking at right there and right there. Right, so the kinetochores forms the site of attachment or bonding, connection, attachments for the sister chromatids. Together, the kinetochores form a structure that we call the centromere. And so the centromere is the region where the two kinetochores, one kinetochore per sister chromatid, form and meet a bond between each other. And so they form that bond that is there. So that's the overall quick and dirty, simplistic view of DNA. So what does this have to do with RNA? And where in the world do we get to start to look at this concept of protein synthesis? Well, that's where we're going next. Right? That is where we're going next. And so let's go ahead and start this. In order to go from DNA to proteins, there are some challenges that needed to be overcome. Right? Challenges. Challenge going from DNA to proteins. First, DNA is isolated to the nucleus of the cell. Second challenge, proteins are produced on the rough ER, on the rough endoplasmic reticulum. Right? And so we have a barrier that is kind of preventing these two structures from even getting together. DNA cannot leave the nucleus. Proteins are only made on the rough endoplasmic reticulum, which is a cellular structure, it's an organelle, that, yes, is attached to the nucleus, but they're separated from one another. There's this nuclear envelope that surrounds the nucleus that prevents the DNA from leaving. And so this presents a major challenge for the body. The information, the blueprints, the directions or instructions for making the protein is locked up in the nucleus found on the DNA. But yet, we need to be able to make the proteins, and that's happening outside of the nucleus. And so what the cell has done is basically created a intermediary host, if you would. And so the solution here was simple. The solution is create an intermediary. Hold on one second. I don't like that. The solution is create an intermediary form of DNA to carry the instructions. And it's exactly what we did. And the body came up with is RNA. So RNA is that intermediary form that is going to be able to be created from the DNA, carry the instructions of the DNA across the nuclear membrane, and deliver it to the site where protein synthesis is going to be happening. Again, protein synthesis. Synthesize, to create. What are we creating? We're creating the proteins. And we're creating the proteins based off of the message that is being carried from the RNA, which originated within the nucleus, being carried on the DNA. And so all of this is starting to hopefully start to piece its way together for us. Now, there's another key aspect here that you really need to understand, and that is something that's referred to as the central dogma of biology. Right? Dogma is a foundational belief. Right? Central means that it is the epicenter of everything that goes on within that particular subject. And so the central foundational building block of biology is found in this relationship where we basically have created a mechanism that allows us to go from DNA to RNA to proteins. everything we discuss within the realm of biology, everything that we discuss in the realm of biology is centered around and is based off of that basic pretense. I can't stress it any more than what I have. This is it. It is the foundation. It is the mother of all building blocks that dictates and drives what happens. If something messes up in any one of these phases, the results can be devastating, or the results could be beneficial. It is when we talk about evolution, evolution is so wrapped around this central dogma of biology. If the DNA is working fine, but somewhere along you alter the strand of RNA, the protein that's produced at the very end is not going to be the protein that is coded for by the DNA. That can cause a mutation, a disease. It could cause something that's beneficial. Everything is based upon that relationship. When we make new antibiotics to fight bacterial infections, those antibiotics are attacking one of three things. They're either attacking the DNA, and if they attack the DNA, they shut the RNA down, which means you can't produce the proteins. Or they're shutting down the RNA. DNA is working fine, but the RNA is messed up. And if you mess the RNA up, the proteins aren't going to be worth anything in the end. Or it goes directly for the proteins. It allows the bacteria or the virus to replicate and make the RNA, and then, bam, it messes the proteins up and it destroys it, breaks it down. It goes ahead and it just completely unravels it. The basis of most of the pills that you pop when you are sick is rooted in the central dogma of biology. Every minute evolutionary change that has happened over the last 3.8 billion years is wrapped around this central dogma of biology. The reason why you have blonde hair and you've got brown hair and you've got black hair and you've got no hair is wrapped around this central dogma of biology. This is it. This is the $1 million answer to the $1 million question. How do we get there? How do we go from DNA to RNA to protein synthesis? How is this all tied together? How is this related to one another? That's where we're going now. First thing you need to understand is that there are three basic types of RNA. We've got mRNA, we've got rRNA, we've got tRNA. These all occur at various stages along the way. And that's part of what we're going to need to look at here. I'm going to tidy up these lines a little bit. That's part of what we need to look at here before we really move on and start looking at anything all too closely. Is what exactly, where are these RNAs being found at? where are these ribonucleic acids functioning, and what impact are they having? So the first one we come up against here is mRNA, which simply stands for messenger RNA. It is carrying the code. Messenger RNA is what is being produced within the nucleus from the DNA. Messenger RNA is being produced within the nucleus from the DNA. And secondly, it is capable of leaving the nucleus. Foundational. All right? And so that's the role of messenger RNA. It's produced within the nucleus, and it's capable of leaving that nucleus and going to the endoplasmic reticulum. Then we have rRNA, which stands for ribosomal RNA. Well, you really probably cannot see that all too well. What happens if I do this? That stinks even more. Right, hold on one second. Switch my color up a little bit. So the second one that we have is ribosomal RNA. The job of ribosomal RNA is simple. To read the message carried on messenger RNA. The job of ribosomal RNA is simply to read the message carried on messenger RNA. And it does that. It does this at the rough ER. Rough ER gets its name. Rough endoplasmic reticulum gets its name because those little rough patches are made up of ribosomes. It's made up of this ribosomal RNA. Now, tRNA stands for transfer RNA. And what transfer RNA does is it takes the message that has been read by rRNA off of the mRNA, and it brings the appropriate amino acid to the game. I'm going to see if I can try to summarize that right here for you. All right, so number one, all right, what does transfer RNA does? It takes the code read by the rRNA from the mRNA and brings, and that's as far as I can go, and it brings the appropriate amino acid to the site. And that right there, once we can start bringing the amino acids to the site, what we've got going on is an amino acid linked to another amino acid via a peptide bond, linked to another amino acid versus another peptide bond, linked to another amino acid versus via a peptide bond, linked to an amino acid, to an amino acid, to amino acid, to an amino acid, to an amino acid, to an amino acid, to an amino acid. And when you've got that long chain of linked amino acids via a peptide bond, what's it called? Primary protein. You've created a primary protein. And so the three of these RNAs must work in tandem with one another. Let's take this another step further. transcription. Let me step back here real quick. Protein synthesis is broken down into two distinct processes. The first of those processes is what we refer to as transcription. Transcription takes place within the nucleus. Transcription takes place within the nucleus. And literally what we are doing is we are taking the DNA and transcribing the code of the DNA into the language of RNA. We're taking the language of DNA and transcribing it into the language of RNA. Specifically, messenger RNA. And so transcription is all about taking DNA and transcribing it into the language of messenger RNA. At that point in time, messenger RNA is permitted to leave the nucleus via a nuclear pore found within the nuclear envelope. And so here is our DNA template. And what we need to create is the messenger RNA chain. So how do we do that? Well, we come in here and we say adenine, when it makes a RNA copy of itself, is going to make, or it's going to bind to thymine is not correct. Remember, we're making a RNA copy of this, and so therefore adenine is going to bind to uracil. And thymine is going to bind to adenine. And guanine is going to bind to cytosine. And guanine is going to bind to cytosine. And adenine, or thymine, is going to bind to adenine. And adenine is going, or adenosine is going to bind to uracil. Cytosine is going to bind to guanine. And guanine is going to bind to cytosine. And cytosine is going to bind to guanine. And adenine is going to bind to uracil. And thymine is going to bind to adenine. What we have just created here, right there, is, for all intensive purposes, a messenger RNA chain. And that messenger RNA chain will go ahead and leave that nucleus and go to the site of protein synthesis on the rough endoplasmic reticulum, which is where we have the second part of the story. Translation. We've transcribed in the nucleus. Now that we're outside of the nucleus, we need to translate into a functional product. And so the key players here during the process of translation are RNA and tRNA. Messenger RNA is going to interact with the rRNA and the tRNA. They're going to interact with one another. It's going to happen. How does all of this go down? Well, a couple things. Right now, this is not going to be any specific order. But I'm going to put together a string of messenger RNA. It's not even going to look like what we just got done talking about. But you'll get the point behind this. U A G A G C U A G G C C U A G G C A U. Well, let's see. Did I do it right? 3, 1, 2, 3. And CG. Okay. Here's my messenger RNA. What ends up happening here is in order for this to be read by a rRNA unit, all right, we've got to be able to break this down into words. This is kind of like when you were first learning to write and you just ran all of your words together in a long sentence and no one could understand what in the world it was you were talking about. Yeah, well, this is kind of what messenger RNA is like. And in order for our RNA to understand what the message is in the messenger RNA, it needs to be able to chop it down into words. Well, words in RNA language are what we call codons. It's a 3-base sequence of messenger RNA. And so if we look, AUG is a codon. That is a 3-base sequence. That is a 3-base sequence. GGC is another codon. CUU is another codon. AGA is another codon. GCU is another codon. AGG is another codon. All right, here's another codon. Here's another codon. There's another codon. And so each of these sets of 3 basically represents a codon. They each represent a different word that the rRNA can understand. And that's what is vital, is being able to break this code down so that the rRNA can understand it. All right, so that's a codon. All right. The anticodon, you see this term anticodon right here, would be a 3-base sequence of nucleotides that are opposite of the codon. And so the opposite of AUG would be UAC. That is the anticodon to the codon AUG. The anticodon for GGC is CCG. The anticodon for CUU is GAA. The anticodon for AGA is UCU. The anticodon for GCA is CGA. GCU is CGA. And so they are the opposites. This would be UCC. This here would be GGA. This here would be UCC. Right, so on and so forth. And this here would be GUU. And this here would be AGC. Right, and so they are the anticodons in green to the codons that were created from the messenger RNA strand. From the messenger RNA strand. The anticodons are contained within the tRNA, the transfer RNA. And as we're going to see here in another slide or so, the tRNA at one end has this anticodon that matches up with the codon on the messenger RNA strand. At the opposite end of the tRNA is an amino acid. And so each amino acid is linked to a specific anticodon, and that anticodon is determined by the codon that is created from the messenger RNA. Now, somebody out there is probably asking me to say that again, so I'm going to try to do that. The anticodon is located on the tRNA, the bottom portion of the tRNA. Opposite of the anticodon, opposite of the opposite end of the tRNA, opposite from the anticodon, there will be an amino acid. And so every amino acid is designated by a certain anticodon. That anticodon matches up with the codon that is derived or created or produced from the messenger RNA strand that we got from transcription, which occurred within the nucleus from the strand of DNA. So we start with DNA in the nucleus, we transcribe the template, we create a messenger RNA strand. That messenger RNA strand leaves the nucleus through the nuclear membrane pore, arrives at the rough endoplasmic reticulum, at which point in time the messenger RNA attaches itself to the rRNA, and the rRNA reads the codon section, these three base sequences of the messenger RNA. As it reads the codon, it then signals for the tRNA with the matching anticodon to come in and bring with it its amino acid. And then it begins to bring in more and more amino acids as this messenger RNA segment moves through the ribosomal RNA. Exciting stuff. One thing I do want to mention is that remember, there are two subunits that make up the rRNA. We have a larger subunit that is known as the 50S ribosome, and we also have a smaller subunit which is known as the 30S ribosome. And we're going to kind of go over that a little bit more in detail in a couple minutes. Let me jump down here to what tRNA looks like. This here is tRNA. It is very often described as a hairpin structure. Okay, it's referred to as a hairpin structure. And you can see that down here at the base is the anticodon, and that anticodon would match up with the codon found on the messenger RNA. And at the opposite end is the amino acid. Right? That there would code for an amino acid. That there would code for an amino acid. Right? So that's what tRNA looks like. Now, when we put this all together, this is what we kind of more or less have. Here is the messenger RNA. You can see that it is moving out of the nucleus through the nuclear pore, and it attaches itself onto the rRNA with its two subunits. This here is the 30S. This here is the 50S. 30S is the smaller subunit, 50S is the larger subunit. It's actually within the 50S where you're going to see the majority of the action take place. And it is down here where we have the tRNA. And again, that tRNA would be brought up into the 50S based upon the codon that was being read by the rRNA contained within the mRNA. So another way of looking at this, I know this is a static view of this. If it was a dynamic view where it was moving, it would make a little bit more sense, and I'm not going to try to solve that here in a couple seconds for you. All right, and so kind of looking at this step by step. All right, step one, right here. All right, we have the DNA found within the nucleus. All right. From that, we create a messenger RNA strand. I lied. All right, sorry about that. From that DNA, we basically separate out a template strand. And so this is the template strand of DNA. How do we know? Because we've got thymine in here. Right? Based off of that template strand, we then create a messenger RNA segment. So this here would be messenger RNA. And you can see it's already broken down into the codons. And each of those codons would then be read by rRNA, to go ahead and determine what the anticodon and therefore the appropriate tRNA with the appropriate amino acid to go ahead and bring in and link together. And so you can see we've got right here, we've got AUG, and that AUG matches up with the anticodon of UAC, and so that's a match. We bring in the amino acid. GCG, the anticodon is CGC, and so that's a match. You bring in that amino acid. GGA, the anticodon would be CCA or CCU, and then you would have its appropriate amino acid down here as well. Right? And you continue to do this until you create that primary structure of the protein. Remember, primary structure of a protein is simply linked amino acids via peptide bonds. And that's exactly what you've got there. Now, from here, what happens? And so in other words, back at step one, we've started in the nucleus. We've gone ahead. We've separated out our DNA. We've made a messenger RNA copy. And step three, that messenger RNA copy is going to leave the nucleus of the membrane. It's going to enter into the rough endoplasmic reticulum, where it's going to meet up with the rRNA. And in step four, the rRNA is going to decipher what those codon regions are, find the appropriate anticodon, bring in the transfer RNA, attached to it would be the amino acid. And in step 5 and 6, those amino acids will get linked together to form the basic primary structure of the protein. Well, what happens from there? There's got to be more to it than just that. And indeed it is. That newly formed protein will get released down into the lumen of the rough ER. All right, so that newly formed protein actually goes down into the rough ER, into the lumen, into this cavity of the rough ER, where it goes it travels to the end of the rough ER. At which point in time, it gets packed up and packaged into a transport vesicle, and it gets shipped to another whatchamacallit, gets shipped to another organelle known as the Golgi body or the Golgi complex. Right? The receiving end of the Golgi body is what we call cis, CIS. All right. So the cis end of the Golgi body receives this transport vesicle from the rough ER containing this primary protein. While in the Golgi body, the Golgi body goes ahead and fixes, if you would, the newly formed protein. So it becomes a secondary protein. It becomes a tertiary protein. It may become a quaternary protein. But while in the Golgi body, it finalizes the protein and it ships it off using here's another new term, secretory vesicle. Right? It's going to be releasing that protein to go wherever it needs to go. The end of the protein that releases the finalized product is what we refer to as trans. So the trans end is what is going to go ahead and utilize the secretory vesicle to release the protein. That's it. That's protein synthesis. Nothing to it. I do want to share with you, if I'm allowed, a short video. I think this will work just fine. At least that's my plan, and I'm sticking to it. So you should have this nice little YouTube video that is popping up on your screen right now. It's only 2 or 3 minutes long, but it's going to review this entire process for us. And so right now we're in the nucleus. And so here we are making the messenger RNA, which is going to be released from the DNA. You'll see it here. It's heading for the nuclear membrane pore. Out of the nucleus goes the messenger RNA, and it's going to go right for our RNA. So now we're doing translation. There's the tRNA with the anticodon. At the opposite end is the amino acid. And out comes that finished protein. That's a primary protein. This here would be the Golgi body. It's known as a chaperone molecule. And so that is protein synthesis, Wimba style. All right, Wimba style. Please spend some time reviewing this. I know this was a little bit longer of a session, but there's a lot of great information in here. And as always, if you have any questions, shoot me an email, let me know, jot them down, ask me during the next lecture or lab. Always here to make sure that you guys get the most out of this as you can. And so until then, happy studying, and don't abuse your proteins too much.